press the bell icon on the YouTube app and never miss another update.
Hello everyone. Well, welcome to the session of the discussion of Neet Grand Paper 12. Well, let us start the discussion from question number 136. I shall be discussing 45 questions for you. Well, to start with, the 136th question for you in front of you. Which one of the following cell organelles is enclosed by a single membrane? So, cell organelles. This is a question coming from the chapter Cell, the structure and function. So, among the given cell organelles are lysosomes, chloroplast, nuclei and mitochondria. So, among them, the single membrane bound structure or the organelle is nothing but the lysosomes. So, as we know that lysosomes are single membraned cells. They are enclosed by a single membrane and inside them they contain nearly 50 hydrolytic enzymes, and the membrane is highly resistant to these enzymes because of high degree of glycosylation, where proteins and lipids deposit, so that the lipids and the carbohydrates they deposit to make the membrane highly resistant against the hydrolytic enzymes which are present in the lysosomes, and hence they are also called as the suicidal. Because they carry the hydrolytic enzymes in them, they are called as hydrolytic enzyme, they are called as suicidal packets. The second one is the chloroplast. As we know, it is a double membranous structure, so which is responsible for photosynthesis, and the nuclei, the one which controls and coordinates all the organelles in a cell, is also double membranous. So mitochondria also double membranous. So outer membrane and inner membrane are present. So even the nuclei is enveloped by the nuclear membrane, which is double membranous. Henceforth, the answer is lysosomes. Well, moving on to the next question. Lack of relaxation between successive stimuli in suspended muscle contraction is known as. So, lack of relaxation during successive stimuli in sustained muscle contraction. That means, so this is a question coming from the chapter Locomotion and Movement. So, contraction and relaxations are brought about during the muscle contraction. So, relaxation between successive stimuli. Stimuli is the messages coming from the brain and towards the muscles is called as the stimuli. So, lack of relaxation between successive stimuli. If there are two stimuli coming over and in between there is continuous contraction without any relaxation, so such a type of condition in sustained muscle contraction is known as, so the answer is tetanus. So, it is called as tetanus. So, because there is lack of relaxation. So, we know that the muscle contraction is brought about by continuous contraction and relaxation, right? So, because of the lack of relaxation between the successive stimuli in the sustained muscle contraction, there is severe pain also brought about, which is called as tetanus. So, it is the continued state of contraction of the muscles. So, this leads to rapid muscle contraction, which is also called as twitching. We call it as twitching. So, the first one is the spasm. Spasm is sudden voluntary contraction of the muscles, which is quite painful. The third is fatigue. Fatigue means undergoing stress or undergoing a lot of tiredness. You call it as fatigue. So, muscles undergo fatigue. So, usually the skeletal muscles, they undergo fatigue. The involuntary muscles like the smooth muscles and the cardiac muscles, they do not undergo fatigue. Whereas the skeletal muscles, which are voluntary in their functions, they undergo fatigue. So, tonus. So, tonus is tension in a muscle which is at rest is called as the tonus, and here also the muscle cannot undergo contraction. So, the muscle cannot undergo contraction in case of tonus. So, lack of relaxation is tetanus, and therefore moving on to the next question. Which of the following is not a stem modification? So the question is coming from morphology of flowering plants. So which is not a stem? There are the examples like tendrils of cucumber, thorns of citrus, picture of nepenthes, and also flattened structures of opuntia. So tendrils of cucumber. So cucumber is nothing but it belongs to the family Cucurbitaceae. So tendrils, they are tiny twisted wire-like structures coming either from the leaflets or coming from the stem. So there are two types of tendrils, stem tendrils and leaf tendrils we call them as. So here there is no modification of this stem. Thorns of so tendrils of cucumber can be or the nothing but stem tendrils. Therefore, this is a stem modification. Thorns of citrus. So thorns are also the modification present on the stem. Therefore, it is also a stem modification. Picture of nepenthes. So nepenthes is an insectivorous plant which looks wherein the leaves are modified to make a picture like a structure or a pot like structure. So the nepenthes leaves are modified into a pot like structure. So the structure looks like this. The picture looks like this, which is just a modification of the leaf. It is just a modification of the leaf wherein it looks like this, and there is one more leaf over this which can be considered as the lid, which can be considered as the lid. So which can close this mouth of the picture. So this is what is called as the picture plant. It is popularly called as the picture plant, or it is also called as the nepenthes. It is also called as nepenthes. So you can see there is modification. This and this are two leaves which are modified into the picture or an insectivorous plant. So this is the cavity. In this cavity, there are certain digestive enzymes. So when insect comes to it and just falls into this, so there are various sticky fluids are present which will trap the insect, and these digestive enzymes will completely digest the insect which gets into the picture. So this is a modification of the leaf but not the stem. Therefore, the answer is picture of nepenthes. So the answer is picture of nepenthes is the right answer. So flattened structures of opuntia. So all of you might have seen opuntia or cactus, wherein the flattened structures are green in color, they are succulent, and they are also responsible for contributing for photosynthesis. So because in cactus, the leaves are modified into thorns. Well, the flattened structures of opuntia are green, succulent, thick, and also contributes for photosynthesis. So and it is the stem modification. Here, the picture of nepenthes is the modification of the leaf. Therefore, picture of nepenthes is the right answer. So moving on to the next question, question number 139. So water soluble pigments found in plant cell vacuoles are. So we know that vacuoles are one of the important organelles. So there are three types of vacuoles. We find sap vacuoles, sap vacuoles, contractile vacuoles, contractile vacuoles, and also food vacuoles. And also food vacuoles. The outermost membrane of the vacuoles is called as the tonoplast. Is called as the tonoplast, and the fluid inside the vacuoles is called as the cell sap. It is called as the cell sap. And this sap is rich in minerals, sugars, amino acids, proteins, minerals, and also certain fat and water soluble pigments. Fat and water soluble pigments. So the water soluble pigments are nothing but anthocyanins. Anthocyanins are nothing but the water soluble pigments which are present in the cell vacuoles. So we have anthocyanins and anthocyanins. Both of them are water soluble pigments, but anthocyanin is not given over here. Only anthocyanin is given. It contributes for red blue color. It contributes for the red blue color, and anthocyanins will contribute for yellow color. Well, this will contribute for red blue color. And carotenoids, chlorophylls and xanthophylls are the part of plastids. They have got nothing to do with vacuoles. So we know, we have studied in detail about the plastids. So they come under the classification of the plastids. Now moving on to the, so therefore the answer is anthocyanins. And now moving to the next question, question number 140. Select the correct statement. So there are four important statements given for us and we need to select the correct one. So the first one, sequoia is one of the tallest trees. So sequoia is a gymnosperm plant. Sequoia is a gymnosperm plant popularly called as giant redwood tree. It is popularly called as giant redwood tree. The full name is Sequoia sempervirens. It is called as Sequoia sempervirens. So it is one of the tallest trees is absolutely correct statement, but still we need to check the other options. The second option tells the leaves of gymnosperms are not well adapted to extremes of climate. So we cannot agree this point we cannot agree because in gymnosperms we find two types of leaves. One is called as the foliage and the other is called as the foliage and the other is the scale leaves. So these foliage, they are of two types. So they might be leathery as in the case of cycas. We have seen in cycas almost they are leathery and they might be needle like in case of conifers. So these are highly resistant to the extremes of the climate. So they are well adapted for the extremes of climate. Therefore, the second statement is absolutely false because it is telling that the gymnosperms are not well adapted. Of course, they are adapted. So the third one is gymnosperms are both homosporous and heterosporous. So it is a wrong statement because gymnosperms are heterosporous. That is, they can synthesis, they can produce two types of spores, that is microspores and megaspores, which contain the male and female gametes responsible for sexual reproduction. So therefore, this is a wrong statement. Then, fourth statement, Salvinia, ginkgo and pinus all are gymnosperms. So, among the given examples, there are three examples given. Salvinia. Salvinia is not a gymnosperm. It is a pteridophyte, aquatic pteridophyte. Ginkgo and pinus are of course gymnosperms. Therefore, this is a wrong statement. So, the correct statement is Sequoia is one of the tallest trees, that is giant redwood tree is the tallest. Well, moving on to the next question. So the question coming from DNA fingerprint technology from molecular basis of inheritance. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? Well, DNA fingerprinting technology is one of the advanced technique to which helps in comparing the two DNA samples. So if the DNA samples, whether it belongs to the same person or to different persons, or whether it belongs to parents and children or they are entirely different, all these conclusions can be drawn with the help of this technology which is called as DNA fingerprint technology. So disputed parentage or with the help of this technology, so various problems related with disputed parentage can be solved. Well, this is one example. Anyhow, which one of the technique is not used here in the DNA fingerprint printing technology is the question. So look into the options: polymerase chain reaction, restriction enzymes, zinc finger analysis, and DNA hybridization. See, during DNA fingerprint technology, so if some robbery has happened or some murder has happened. So someone is suspect of have committing this murder. And the DNA sample which is available at the spot of the where the murder has taken place there, and the DNA sample of the suspected victim are compared. So using this technology. So what they do is whatever the sample is got, maybe it is saliva or semen or such another or such a, even hair follicles, if they are got, so DNA is extracted from that. They are there, the amplification of the DNA, the quantity of that DNA is increased with the help of polymerase chain reaction, which we call it as amplification of the desired gene. So this is of course one of the technique that is used in DNA fingerprinting. So second one is restriction enzymes. So restriction enzymes are those enzymes which cut the DNA fragment at specific sites or specific sequences. Then zinc finger analysis. As such, zinc finger analysis is not used in DNA fingerprint. So zinc fingers are nothing but the proteins that are available in the eukaryotic genome. So they are nothing but the factors or the proteins that are available in the eukaryotic gene, we can tell. So zinc finger analysis is not at all used. DNA hybridization, of course, the DNA of the sample and the victim, both of them are compared for DNA hybridization is done. So for comparing the both DNA, and this method we call it as southern blotting, and which is done with the help of DNA probes. Well, these polymerase chain reaction is a technique which is used under DNA fingerprinting. Restriction enzymes are required for DNA fingerprinting. Zinc finger analysis is not required. DNA hybridization is required. So the one that is not required is zinc finger analysis. Yes, moving on to the next question. Well, the question is, which of the which type of tissue correctly matches with its location is the question. So we have four options. Yeah, tissue and its location. In the first, we have areolar tissue, and the example that is given over there is tendons. Yes, so areolar connective tissue is a loose connective tissue, whereas tendons is not a loose connective tissue, but it is dense connective tissue. So it is a mismatch, I hope. So tendons are nothing but the dense regular connective tissues, and they are which are rich in white fibrous tissue. That is, they are made up of collagen fibers. Whereas areolar connective tissue is a loose connective tissue which contains huge amount of matrix and the fibers and the various types of cells suspended in it. In the second case, the tissue is smooth muscle and the location is wall of intestine. So I think this is a right match because smooth muscles are involuntary muscles and the walls of the intestine are also involuntary in function. Therefore, this is a right match. But still look into the third and fourth option. So the third option is cuboidal epithelium lining of stomach. So the lining of the stomach is brought about by the columnar epithelium but not cuboidal epithelium. So cuboidal epithelium are present in the ducts of the glands. So the fourth one is transitional epithelium and it is present at the tip of the nose. So this is also a mismatch because transitional epithelium, they are present in the renal pelvis. They are also called as urothelium because most of it is present in the urinary part of the body, that is in the uterus, the urinary bladder and also the urethra. So tip of the nose consists of cartilage but not transitional epithelium. So the right answer is this option 2, smooth muscle wall of intestine. Well, moving on to the next question. Question number 143. Well, a plant in your garden avoids photorespiratory losses, has improved water use efficiency, shows high rates of photosynthesis at high temperatures, and has improved efficiency of nitrogen utilization. And in which of the following physiological groups would you assign this plant? So there are list of um, plants given over there: CAM plants, C4 plants, C3 plants, and also nitrogen fixing plants. So among these four types of plants, so these qualities like avoiding the photorespiratory losses and improved use of water efficiency and high rate of photosynthesis at high temperature. All these can be observed in C4 plants. We can all observe this in the C4 plants. So the C4 plants are the one which will undergo the Hath and slack cycle during its photosynthesis, right? So here the first product formed is oxaloacetic acid, which is a 4-carbon atom. Hence, they are called as C4 plants. And the reaction, the optimum temperature required for the reaction is the reactions can occur in even 30 degree to 45 degree. That is, high rate of photosynthesis can be seen at high temperatures where there is what nearly carbon dioxide, 40 to 80 milligrams of carbon dioxide decimeter square per hour is utilized. So CAM plants. So CAM plants are also a type of plants which will follow the C4 pathway, but the speciality of these CAM plants is, so the full form of CAM is crassulacean acid metabolism, we call it as, and here in this the speciality of these organisms, this type of plants is the carbon dioxide is taken up at night when the stomata are open and fixed into malic acid. So the carbon dioxide that is accepted is converted into malic acid, and in the daytime when the stomata is closed, carbon dioxide is released by malic acid, and that carbon dioxide is used for Calvin cycle in C4 plants. So C4 pathway only, a little deviation from the C4 pathway we can see in CAM plants, and some of the examples or some of the xerophytes, like the best example is pineapple. Then nitrogen fixing, we know we have a group of plants which helps in nitrogen fixation like the rhizobium or the pseudomonas or etc. So C3 plants, the one which follows the C3 pathway, where the first product formed is PEP, which is the first product that is formed is the PGA, which is a 3-carbon atom compound. Well, moving on to the next question. Which of the following structures is homologous to the wing of a bird? So, homologous structures. This is a question coming from the chapter evolution. So, in evolution, we have studied homologous structures and analogous structures. So, homologous structures are the structures which are present in different organisms which are similar in their origin and structure but perform different functions. So, wing of a bird, the origin of the wing of a bird and the flipper of whale both are same, but the functions are different. So the wing of the bird helps in flying, whereas flipper of whale helps in swimming. Well, therefore, these two can be called as the homologous organs. So the answer is flipper of whale. The first one is the hind limb of rabbit. So hind limb of rabbit has got a different structure when compared to the wing of a bird. So wing of a moth and wing of a bird can be analogous organs but not homologous organs. Then dorsal fin of a shark. So the dorsal fin of a shark cannot be a homologous organ, though it helps in locomotion, but it helps here. Dorsal fin of a shark helps in balancing of the, it acts like a balancing organ where the rolling of the shark is prevented with the help of the dorsal fin in case of shark. Well, therefore, the right answer is flipper of whale is homologous to the wing of a bird, which explains homology, an important phenomenon telling that these structures have developed from these organisms might have got a common ancestor, which is Also an example for divergent evolution. Well, moving on to the next question. Which of the following characteristic features always holds true for the corresponding group of animals? So, in every condition, this character should hold good for the particular group of animals. So, let us look into such example. So, which is the right answer? So, viviparous mammalia. Can this example be a characteristic in every always true? Is it always true? No, mammals, we have various examples of mammals which are also egg laying. Like the platypus and the echidna are egg laying, they are oviparous but they are not viviparous. So, this is not true always. In the second case, possesses a mouth with an upper and a lower jaw, chordata. So, all chordates do not have a mouth with an upper and a lower jaw. So that is only the agnathans do not have upper jaw and lower jaw. Only the gnathostomata have the upper and lower jaw. So under chordates we also have the cyclostomes. Therefore, this will not hold good. Yes, looking into third example, cartilaginous endoskeleton chondrichthyes. So chondrichthyes is nothing but a group of fishes which have got an endoskeleton which is made up of cartilage. So I think this will hold good always and therefore this can be the right answer. In the fourth case, let us check it out also. A three-chambered heart with one incompletely divided ventricle is seen in reptiles except in crocodiles because crocodiles are having four-chambered heart. Therefore, this is not always true. Therefore, this cannot be the answer. Moving on to the therefore third option, cartilaginous endoskeleton chondrichthyes is the right answer. Moving on to the next question. Which of the following statements is not true for cancer cells in relation to mutations? So, mutations we know due to mutations, body cells, normal cells can be converted into uh, true cells. The normal cells can be converted into cancer cells due to mutations. And something which is related to mutations is given in four important statements. We need to check out which is wrong, right? So the first one tells mutations destroy telomerase inhibitor. So second one tells mutations inhibit production of telomerase. Third, mutations in proto-oncogenes accelerate the cell cycle. Mutations inactive the cell control. Well, which is not true for cancer cells in relation to mutations. The first one is mutations destroy telomerase inhibitor. So this telomerase activity goes on increasing in the cancer cells and mutations destroy telomerase inhibitor is of course uh, the right statement. So it will destroy the telomerase inhibitor. So telomerases are nothing but the enzymes which will go on adding certain bases at the 3' end of the genetic material, right? So therefore the telomerase activity goes on increasing in case of cancer cells. So which brings about the increased cell division, So cancer cells, there is increased telomeric activity or the telomerase activity and also there is an increased cell division. So mutations destroy telomerase inhibitor is absolutely right. Second one, mutations inhibit production of telomerase. So this is true. That is, it is not a statement regarding the mutations, about mutations. So mutations inhibit production of telomerase is wrong. Instead, mutations disable the telomerases from doing its function. So mutations inhibit production production irutte, but they stop the functions temporarily. So mutations in proto-oncogenes accelerate the cell cycle is absolutely true statement. Mutations inactive the cell control. Yes, the cell control system has been hampered, which has brought about increased cell division activity. Therefore, the mutations except mutations inhibit production of telomerase. All the other statements are absolutely right. Next, the amino acid tryptophan is the precursor for the synthesis of. Well, amino acid tryptophan is the precursor of synthesis. So this question is related with chemical coordination and integration chapter, where we have studied about the protein hormones which are made up of proteins, hormones which are made up of steroids. So like this, and we also have iodohormones. So now the question is, which are those hormones which are made up of tryptophan is the question. So this is simple common sense we need to use to analyze this question. See, amino acid tryptophan means they are only present in protein hormones, right? So with this basic thing, let us look into the options. So the first part option is thyroxine and triiodothyronine. So thyroxine and triiodothyronine are iodohormones, but they are not protein hormones. Therefore, first option cannot be the answer. In the second case, estrogen and progesterone. So both estrogen and progesterone are steroid hormones and they are not made up of proteins. Therefore, even tryptophan may not be present there. So the third option is melatonin and serotonin. So melatonin and serotonin, they can be the right answer because they are hormones, protein hormones, because they are protein hormones. So all the hormones which are secreted from the pituitary gland can be are all made up of proteins, and melatonin is one such hormone which is secreted from the pituitary gland and it is made up of proteins. Cortisol and cortisone, of course, they are also peptide hormones which are made up of amines but not amino acids. Therefore, the right answer is melatonin and serotonin. Moving on to the next question. Following are the two statements regarding the origin of life. The earliest organisms that appeared on the earth were non-green and presumably anaerobes. The first autotrophic organisms were the chemoautotrophs that never released oxygen. Of the above statements, which one of the following options is correct? So we need to analyze this question from point of evolution. So we have studied in evolution, we have studied something about the origin of life. So the origin of life has been successfully explained with the theory which has been given by A.I. Oparin and J.B.S. Haldane, which is called as the chemical theory of origin of life. So according to this, the prebiotic earth was made up of, that is the primitive earth, that is during the before the origin of the first life, the prebiotic earth was simply made up of certain atoms like carbon, oxygen, nitrogen and also hydrogen, which combine together to form the simple molecules like carbon dioxide, H2O, then hydrogen cyanide, NH3 and also CH4. So they got converted into simple molecules. They got converted into simple molecules. So these simple molecules further became organic molecules and they underwent polymerization to form macromolecules. They formed the macromolecules, and these macromolecules, so the mixture of the macromolecules or the mixture of the proteins, they started showing certain characteristics of the first growth. Therefore, they were called as protobionts. They were called as the protobionts. And from the protobionts came the ebionts. Ebionts were the first living cells, we can tell. So from the ebionts came out the first prokaryotic life, which were initially heterotrophic. So the one which were formed were heterotrophic, and then we got the autotrophic ones, and later came the development of the autotrophic organisms, and finally we got the eukaryotes. Finally, we got the eukaryotes. Well, with this basic information, let us compare the two statements and finally analyze the answer. Excuse me. The earliest organisms that appeared on the earth were non-green, that is, they are non-photosynthetic and presumably anaerobes. So, of course, the first formed earliest animals were anaerobes. They never used oxygen and they were non-green. So, that is, we can tell them as protobionts. We can call them as the protobionts. The first autotrophic organisms were the chemoautotropes. Yes, they prepared their own food or synthesized their own food with the help of simple molecules, chemical molecules, and never released oxygen. Of the above statements, which one of the following options is correct? So, both the options A and B are absolutely correct according to evolution point of view, what we have studied. So, moving on to the next question, question number 149. Reduction in pH of blood will so, what reduction, if there is a reduction in the pH of the blood? So, what is exactly the pH of blood? So, the pH of blood is 7.4, which is almost neutral. And whenever the blood moves towards the blood near the lungs, when the blood is near the lungs, so there is an increased concentration of carbon dioxide, which increases the pH level. So, increased concentration of carbon dioxide will increase pH is what we have studied in our syllabus. So, reduction in pH of blood will obviously reduce what? So, it will decrease the affinity of hemoglobin with oxygen. So, when the blood is moving towards the oxygen, towards the lungs means, so at that time there is next process will be oxygenation, where there will be an increased affinity for oxygen by the hemoglobin. Now, because there is the concentration of the carbon dioxide is more, the pH is also increased at that time. Now, the think in a reverse way, when there is reduction in the pH, obviously there is a decreased affinity of hemoglobin with the oxygen will be the right answer. So, reduction of pH of blood will decrease the affinity of hemoglobin with oxygen. So, reduction in pH shows that there is reduction in carbon dioxide. So, reduction in carbon dioxide tells that there is an increased concentration of oxygen. So, when there is already oxygen present in the blood, so there is less affinity for hemoglobin with the oxygen. So, with this analysis, we will come to the conclusion that decrease the affinity of hemoglobin with oxygen is the right answer. So, moving on to the next question. Analogous structures are a result of analogous structures. So, both analogous structures and homologous structures are evidences of evolution. We have studied in detail. So, analogous structures. So, they are dissimilar in the structure, but they have common functions. So, such structures can be called as analogous structures. So, the best example can be eye of octopus and man. Both have similar function, but their origin is quite different. So, you can also take another example, that is potato and sweet potato. Both of them, they have common functions like storage of food, but they have different structures. Potato is stem modification, whereas sweet potato is the root modification. So, here the structures have a common purpose, but they have different they have a same purpose, that is the functions are same, but they are developed from different parts because they might have come from different habitats, so it is what you call it as analogy. They might have got some shared ancestors, so analogous or structures are an example for convergent evolution. So, convergent evolution. So, divergent evolution is brought about by homologous organs, where organs develop in different direction for the according to the needs of the organism. Stabilizing selection. So stabilizing selection is a type of natural selection. So we have studied stabilizing the selection, directional and also disruptive. Three types of natural selection we have studied. So stabilizing selection. So usually the nature is nature favors the average ones, whereas the individuals which are at the extremes are totally removed. So the stabilizing selection we call it as. And finally it is shared ancestry, which we have discussed in convergent evolution. So analogous structure is the direct result is convergent evolution. So moving on to the next question, question number 151. Which of the following is a restriction endonuclease? So restriction endonucleases we have come across in biotechnology. So restriction endonucleases are also called as molecular scissors because they are used to cut the DNA strands at specific sites. Now the question is, which of the following is a restriction endonuclease? So the best example for the restriction endonuclease is the HIND2. So we have in detail studied the PUC 322 plasmid, so which consists of HIND2 as the restriction endonuclease. So the first option, DNase. So DNase 1 is an enzyme which can digest only DNA. RNase or enzymes which can digest RNAs, and protease or those enzymes which can digest proteins. Well, moving on to the next question, question number 152. The term ecosystem was coined by, well, the word ecosystem. So ecosystem can be called as the unit of ecology. So wherein we find biotic and abiotic factors interacting with each other, whereas there is lot of energy production, there is lot of productivity, lot of functions like decomposition and the things going on. So this term ecosystem was coined by A.G. Tansley. So A.G. Tansley coined this term ecosystem in the year 1935. And E. Haeckel is related with coining the word ecology in 1869. The third is E. Warming. So E. Warming was a Danish botanist. He wrote the first textbook of plant ecology. And finally, we have E. P. Odum, so who is also called as Eugene Pleasance Odum, who is called as the father of modern ecology. Next, moving on to the next question, question number 153. Which one of the following statements is wrong? So we need to check out all the four statements and tell which is wrong. First one, cellulose is a polysaccharide. Of course, so cellulose is made up of, is a type of carbohydrate. So carbohydrates have been broadly classified into monosaccharides, oligosaccharides and polysaccharides. So polysaccharides which are made up of many sugar molecules. So sugars are the monomers. So they are polymerized, hence called as polysaccharide. And cellulose is called as a homopolysaccharide because it is made up of many glucose molecules. Well, uracil is a pyrimidine. So you need to find out the wrong statement. So of course uracil is a pyrimidine. Glycine is a sulfur containing amino acid. So glycine is not a sulfur containing amino acid. So you might have come across the structure of glycine in the chapter biomolecules. So we know the structure of glycine. So wherein it is an amino acid containing a variable group. So C and H2, H, COOH, and here we have H. So this is the variable group in any amino acid. So if it is H, you call it as glycine. We call it as glycine, and glycine do not contain any sulfur. Here we can see the structure of glycine. So this is glycine. So this is the generalized structure of amino acid, where this is the variable region and can be called as R. So for different amino acids, we have different R. For different amino acids, we have varying R. The R goes on varying. R goes on varying, and here the R, the variable part consists of one hydrogen which is called as glycine. If it is replaced by CH3, you call it as alanine, and if it is replaced by CH2OH, you call it as serine. We call it as serine. So glycine is not a amino acid which contains sulfur. Instead, methionine and cysteine, methionine and cysteine or those amino acids which contain sulfur in them, which contain sulfur in them. Well, moving on to the next question, question number, and the last one is sucrose is a disaccharide. So the first, second and the fourth statement are absolutely right, whereas the third is wrong. Therefore, the answer is option 3. Well, looking into the next question, question number 154. In bryophytes and pteridophytes, transport of male gametes requires. Answer is water, because both these gametes are flagellated gametes. So they are motile gametes, and for their movement they require water. Therefore, they are motile. Since the gametes which are produced by bryophytes and pteridophytes are motile or flagellated gametes, they need water for reaching the female gamete. Yes, moving on to the next question. When does the growth rate of a population following the logistic growth model equal to 0? The logistic model is given as dn by dt is equal to Rn into 1 minus n by k. Yes, so the logistic growth model. In the chapter called as organisms and population, we have studied two important growth models. One is called as the exponential growth model, and the other is called as the logistic growth model, which has been given by Ernst Pearl. Right? So here in this, according to this model, dn by dt, population density, is equal to Rn divided by k minus n divided by k is the equation for logistic growth model. So k minus n divided by k. So where R is called as the intrinsic rate of increase, which is also called as the maximum reproductive capacity, which can be called as the maximum reproductive capacity. Now, the demand is, when does the growth rate of a population becomes 0? Yes, this is the question. So, when it should become 0? If the value of k minus n by k is 0, if this value is 0, which is multiplied by Rn, then it will obviously become 0. So, if the value of when n by k equals 0, so the first, when death rate is greater than birth rate, when n by k equals 0, when n nears the carrying capacity of the habitat, and but in that equation they have given Rn divided by 1 minus nk. In this equation you have to apply our knowledge to this equation and tell when this will become 0, right? So now this can become 0 only when there is 1 minus 1, right? So when 1 minus 1, that is when n by k is equal to 1. When the value of n by k is exactly 1, so the growth model, that is the logistic growth model, will be equal to 0. Therefore, the right answer is when n by k is exactly 1, the growth model will become 0. Moving on to the next question, we need to look into the statements and find out the correct statement. So, true statement we need to fetch out. Well, the question is about anther, that is the male part of the flowering plant. Which one of the following statements is not true? So, exine of pollen grains is made up of sporopollenin. So, we do agree that exine of pollen grains is made up of sporopollenin. sporopollenin, which is highly resistant. Second one, so it is a true statement. We need to fetch out the wrong statement. The second one is pollen grains of many species can cause severe allergies. Is absolutely true. So various type of allergies like the rhinitis, asthma, bronchitis are all caused because of the pollen grains. And some of the plants that contribute for allergy or the parthenium or the common grass or even the inflorescence of the sorghum vulgare, all these are some of the plants that contribute for allergies. The pollen grains of these plants will cause allergy. So stored pollen. So option 2 is also a right statement. Third one, stored pollen in liquid nitrogen can be used in the crop breeding programs. So we have studied about cryopreservation. Of course, these pollen grains can be preserved and can be used for crop breeding programs when they are required. And finally, it is tapetum helps in the dehiscence of anther. So we know the structure of the anther, a bilobed anther and a unilobed anther. So a bilobed anther will have four sporangium structures, and in them we find this tapetum. We find the tapetum. So this tapetum helps in nourishing. It provides the nourishment for the development of the pollen grains. So but it is not going to contribute for dehiscence of the anther. So dehiscence of the anther is brought about by the degeneration of the middle layers in the anther wall. Yes, therefore option 4 is the wrong statement. And moving on to the next question. Which of the following would appear as the pioneer organisms on bare rocks? So we have studied about something called as succession in the chapter ecosystem. Succession. So succession is something where a entire biotic community has developed to its maximum in a particular place where there was nothing once. So this may take, so this succession is of two types, primary succession and the secondary succession. So the primary succession, so for the development of an complete biotic community may take nearly thousands of years. So there's also secondary succession, ecological succession, which may take nearly hundreds of years for the development of the biotic community. Whatever it is, which of the following would appear as the pioneer organism? So when nothing is there, so on a bare land, the first organism that develops on a bare place or a place where there was nothing, no vegetation. So the first organism that develops is called as the pioneer. And the question is the pioneer organisms on bare rocks. So you call this as zero seed. We call this as zero seed, where biological community is going to develop on the bare rocks, and the pioneer species or the pioneer organisms is nothing but the lichens. So lichens are the first organisms which are going to develop on the bark of on the bare rocks. So which is also followed by mosses. So the pioneer species or the pioneer organisms are the lichens, and which is followed by the mosses, which is followed by the grasses, and which is followed by the perennial grass, then the shrubs, and finally the small shrubs. So this is how there is a predictable change that is going on in a succession. Yes, so the pioneer species here is lichens. So moving on to the next question. Which one of the following is the starter codon? So the starter codon is AUG. AUG is called as the starter codon, or it also codes for the amino acid called as methionine. So this has got two functions. AUG is a codon which has got dual function. So AUG stands as a start codon, and also it acts like it synthesizes the amino acid. Sorry, it represents the codon methionine. So UGA, UAG and UAA, all of them are called as nonsense codons, or they are also called as terminator codons. So which stop the transcription. So they are called as stop codons or terminator codons. So moving on to the next question. Question number 159. Which one of the following characteristics is not shared by birds and mammals? So we have some common functions, common characteristics of birds and mammals. So which are those characters that are not shared by birds, which are not common to birds and mammals is the question. So both of them are warm-blooded in nature is absolutely right. Both of them are homeothermic, that is, their body temperature remains a constant irrespective of the external temperature. The second is viviparity. So viviparity is giving birth to young ones. So viviparity can be seen in placental mammals and marsupial mammals, but not in case of egg-laying mammals. So this viviparity is not at all a characteristic of birds. All birds are exhibit oviparity where they lay eggs. Therefore, viviparity is the character which is not shared by birds and mammals, we can tell. But still, cross check whether the other two also do not belong to the answer group. So, breathing using lungs. Of course, both mammals and birds possess lungs, which helps in breathing. And ossified endoskeleton. Yes, ossified endoskeleton is the endoskeleton is made up of bones. You call it as ossified endoskeleton. Of course, both of them have ossified endoskeleton. Therefore, the answer is option 2, viviparity. Moving on to the next question, question number 160. Nomenclature is governed by certain universal rules. Which one of the following is contrary to the rules of nomenclature? So, in the biological classification, we have studied how organisms can be classified and how organisms can be named with the help of scientific names. So, nomenclature is governed by certain universal rules which has been done at the international levels by the ICBN. So International Code for Botanical Nomenclature, so has framed rules. Which one of the following is contrary to the rules of nomenclature? Contrary means that the one which is opposite to the rules, which is against the rules of the one which is given at the international level is the question. So biological names can be written in any language. So I think this itself is the one which is against the universal rule. The universal rule tells biological names should be in Latin. So second, when written by hand, the names are to be underlined, of course. So Homo sapien, if we write Homo sapien, the first letter of the generic name is H, which is written in capital, and the specific name Sapien, the initial letter is to be written in small letter and they should be underlined separately. The names are written in Latin and italicized. Yes, of course, they are written in, they originate from the Latin language. The first word in a biological name represents the genus name, and the second is a specific epithet, is also absolutely right. So, something which is against the rules framed by the ICBN is biological names can be written in any language. Therefore, option 1 is the right answer. Moving on to next question, question number 161. Blood pressure in the pulmonary artery is. So, in the pulmonary artery, what is the blood pressure? Whether it is more than that in the carotid artery, whether it is less than that in the vena cava, more than that in the pulmonary vein, or same as that in the aorta. So, pulmonary artery, as we know, carries the blood from the right ventricles towards the two lungs. So, it contains deoxygenated blood. So, in the pulmonary artery, when you look into the structure or the section of the pulmonary artery, so the pulmonary artery has got, this is the section of a pulmonary artery. So, you have the lumen is very small. So this is the lumen. This is the lumen of the artery, which is very small, and this is the layer of smooth muscles. This is the wall, this is the smooth muscles, and this is the lumen. This is called as the lumen. And when you compare with the veins have a bigger lumen. They have got a bigger lumen when compared to the artery, and this is the layer of smooth muscles. This is the lumen. Now, when the lumen is bigger, the pressure is less, right? When the lumen is bigger, the pressure is less, and when the lumen is smaller, the pressure is more. So, Boyle's law tells both are inversely proportional. So, blood pressure in the pulmonary artery is more than that in the pulmonary vein. So, this is the pulmonary vein where the lumen is big and here the lumen is small, and therefore the um, blood pressure is less in pulmonary artery and more in pulmonary vein. Yes, that is option 3 is the right answer. Moving on to the next question. 162. So use some simple logic to answer this question, because we know something about the lumen of the artery and the vein. We have studied with the help of the diagram. Other things are not mentioned, much of it. So using simple logic, you can answer this question. So moving on to the next question, question number 162. Cotyledon of maize grain is called as scutellum. So we have the scutellum. So we have the aleuron layer. We have the scutellum. So you will have the aleuron layer. So we have the aleuron layer, which has got an embryonal axis, which comprises of the radicle and the plumule, right, like this. So this is the aleuron layer. Yeah, I have written a very simplified diagram. So we have the radicle and the plumule. So radicle, so this is plumule, which develops into a shoot system, and this is radicle, which is going to develop into a root system, and this is the coleoptile. This is the coleoptile, which is a protective sheath, and this is the scutellum, and this is called as the scutellum, which is the cotyledon part, which is the cotyledon part. So the cotyledon of maize grain is nothing but the scutellum. Yeah, moving on to the next question. In the stomach, gastric acid is secreted by the acidic cells, parietal cells, gastrin secreting cells, and the peptic cells. Gastric acid, that is HCl, is secreted from the parietal cells of the gastric glands, not the acidic cells, not the gastrin secreting cells, also called as G cells, which are neuroendocrine and present in the pyloric part of the stomach and also in the duodenum and pancreas. So peptic cells, so they are going to secrete the zymogens like the pepsinogen and prorenin, and acidic cells. So usually acidic cells, so is a word used with the pretext of cancer cells, where the pH of the cancer cells, because it is high, hence it is called as acidic cells. So the one which produces the HCl is the parietal cells. So moving on to the next question, question number 164. Depletion of which gas in the atmosphere can lead to an increased incidence of skin cancers. So skin cancers are brought about by depletion of which gas. So we know the atmosphere consists of the different layers, and among them is the stratosphere. This stratosphere consists of ozone, O3 compound, which O3, which is capable of trapping the absorbing the UV radiations, harmful UV radiations coming from the sun. So this where due to the depletion of this ozone, there are some poisonous, harmful rays entering onto the planet Earth and destroying the various organisms. And one such incidence due to decreased ozone is that is skin cancer. So usually in the Antarctic region, there are so many holes which are created in the winter where there is depletion of the ozone layer. And the depletion of ozone layer is due to certain ozone depleting substances. And one of the substances is the nitrous oxide or the chlorofluorocarbons, which are going to deplete the ozones. So when ozone is the thickness of the ozone is decreased or there is no ozone, creating a patch or a hole which is called as ozone hole. So through this ozone hole, the UV radiations reach the earth and cause mutations in the cells of the organisms. So that mutations might lead to various skin cancers in human beings. So the answer is ozone. So moving on to the next question, question number 165. Chrysophytes, euglenoids, dinoflagellates and slime molds are included in the kingdom. Options are fungi, monera, animalia and protista. So the right answer is protista. So chrysophytes, euglenoids, dinoflagellates, slime molds are all examples of protistans. So Chrysophyta, we have the diatoms. Euglenoids, we have euglena itself as the example. Dinoflagellates, we have goniolox as the example. And Plasmodium, we call it as protean fungi. Fuligo is one of the example. So all these are protistants. So fungi is once again divided into various classes like the Phycomycetes, Ascomycetes, Deuteromycetes and Basidiomycetes. So monarans have archaebacteria, then also they have the eubacteria and also blue-green algae along with the mycoplasma. All of them come under monarans. Animalia, we have the several groups starting from the poriferans to chordates. We have various groups of animals which come under animalia. Therefore, chrysophytes, euglenoids, dinoflagellates and slime molds are nothing but protistants. Moving on to the next question. Water vapor comes out from the plant leaf through the stomatal opening. Through the stomatal opening, carbon dioxide diffuses into the plant during photosynthesis. Reason out the above statements using one of the following options. So we need to fetch out the reason for those two statements in the given options. So now looking into the options. One process occurs during daytime and the other at night. Both processes cannot happen simultaneously. The above processes happen only during night. So both processes can happen together because the diffusion coefficient of water and carbon dioxide is different. So absolutely the answer is both processes can happen together because the diffusion coefficient of water and carbon dioxide is different. So water vapor comes out from the plant leaf, that is you can tell it is transpiration. Then through the same stomatal opening, carbon dioxide diffuses into the plant for photosynthesis. So both can happen simultaneously because the stomata is open and both have different diffusion coefficients. Well, moving on to the next question. In mammals, which blood vessel would normally carry largest amount of urea? So the question is related to urea. So urea is formed by ammonia with the help of a cycle which is called as the ornithine cycle, which occurs in the liver. In the liver occurs a cycle which is called as ornithine cycle, so through which the ammonia is converted into urea, the more toxic substance. So this moves uh, so this urea is present in the deoxygenated blood, and the one which carries the deoxygenated blood is the hepatic vein. So the hepatic vein is the one which carries the deoxygenated blood and is rich in ammonia. So hepatic portal vein is the veins connecting the intestine and the liver. Hepatic portal vein we call it as. So we know what is aorta, the one which starts at the base of the left ventricles and branches into various arteries and arterioles. Renal vein, so the vein of the kidney, we call it as the renal vein, which carries the blood away from the kidneys. Well, moving on to the next question. Seed formation without fertilization in flowering plants involves the process of. So the right answer is apomixis. So it which forms apomictic seeds, which is due to which are formed without fertilization. So somatic hybridization, we know that the protoplasm of two different cells, plant cells, can be fused together, which is called as somatic hybridization, and the products like pomato, bomato are all produced by somatic hybridization. So the second one is sporulation, that is the formation of the spores is one of the process under uh, asexual reproduction, which can be seen in Monera protestans and also fungi and also even algae. Then budding. So budding is also a type of asexual reproduction that is seen in hydra and also in yeast. Well, that is about this question. The answer is apomixis. Moving on to the next question. 170. 169. Which of the following is wrongly matched? Which of the following is wrongly matched in the given table? So there are four different uh, microbes along with their product and application. We need to see which was the one which wrongly matched. We need to fetch out the one which is wrongly matched. The first one is Clostridium botulism. Product is lipase and application is removal of oil stains. We can agree with the product and its application, but not with the microbe that produces lipase. So, Clostridium botulism is used in the production of butyric acid rather than lipases. So, lipases are extracted from Candida lipolyticum and also Geotrichum candidum, but not from Clostridium botulism. Therefore, this itself is the wrong option. And the second, just check out whether second, third, and fourth are absolutely right. So second is Monascus purpureus. The product which is extracted from it is statins, and the function is it functions as lowering the blood cholesterol lowering agent is absolutely right. In the third option, we have Streptococcus bacteria. The product we get is streptokinase, which is used in the removal of clot from blood vessel, is also rightly matched. Fourth one is microbe, Trichoderma polysporum. Product is cyclosporin A and the application. So it is used as a immunosuppressive drug is also absolutely rightly matched. And therefore moving on to the next question, question number 170. Well, we have a question coming from principles of inheritance and variation. So, in a test cross involving F1 dihybrids, so remember it is a test cross and it is involving F1 dihybrid flies. More parental type of spring were produced than the recombinant type of spring. This indicates, so it is a test cross F1 dihybrid which is crossed with the recessive parent. So then what is happening? More parental type of offsprings were produced rather than the recombinant type of offspring. So this indicates, look at the four options, both of the characters are controlled by more than one gene. If the two characters are controlled by more than one gene, you call it as, we call it as um, multiple allelism. The second one, the two genes are located on two different chromosomes. The third is the two genes are linked on the or present on linked or present on the same chromosomes. And finally, chromosomes fail to separate during meiosis. So the two genes are located on different chromosomes cannot be the answer. The two genes which are linked on the same chromosome but can cause recombination, and also we get parental type of offsprings. The two genes are located on the same chromosome, option 3. So when they are located on the same chromosome, there is all possibility that the offsprings are of two kinds, so parental type and also recombinants. So less of recombinants if the genes are tightly linked. If they are loosely linked, there will be more type of recombinant, and more than the recombinants will be the parental type of organisms. So the fourth one, chromosomes fail to separate during meiosis. So this is a condition called as polyploidy. When there is a chromosome fail to separate during meiosis. So what happens? The chromosomes remain together, therefore the number of chromosomes becomes more in the, there will be a double set of chromosomes formed, which you call it as polyploidy condition. We call it as polyploidy condition. Therefore the right answer is option 3. Moving on to the next question, question number 171. It is much easier for a small animal to run uphill than for a large animal because the efficiency of muscles in large animals is less than in the small animals. Smaller animals have a higher metabolic rate. Small animals have a lower oxygen requirement. It is easier to carry a small body weight. So, well, we have to answer this with a scientific background. Therefore, looking to the options, the efficiency of muscles in large animals is less than in the small animals. We cannot agree with this answer. So, the efficiency of muscles is very greater in huge animals also. The smaller animals have a higher metabolic rate. So this is, we can agree with this because metabolic rate is directly proportional to the size of an animal. So larger the animal, lesser is the metabolic rate, and smaller is the animal, higher is the metabolic rate. Therefore, smaller animals have a higher metabolic rate. Therefore, they can move uphill or run uphill faster than a larger animal. So this also holds good with human beings also. The man who is very strong and thin will move very fastly on a, when he has to uh, go on the staircase, he will move faster, whereas a person with more weight moves very slowly. So the metabolic rate, here it is the question of metabolic rate. The metabolic rate is directly proportional to the size of the animal. More is the animal size, lesser is the metabolic rate. Smaller is the animal, more is the metabolic rate. Therefore, the answer is smaller animals have a higher metabolic rate. Moving on to the next question, question number 172. Which of the following is not a characteristic feature during mitosis in somatic cells? So, mitosis and meiosis are two different types of cell divisions. So, mitosis takes place in somatic cells, whereas meiosis takes place in germ cells. And now, which of the characteristics is not present in mitosis is the question. Look into the options. Chromosome movement, option 1, chromosome movement. Second, spindle fibers. 3, disappearance of nucleolus. And fourth is synapses. So chromosome movement can be seen both in mitosis and also meiosis. Spindle fibers are also formed both in mitosis and also meiosis. Disappearance of nucleolus and its reappearance is also found in both mitosis and meiosis. But synapsis is found only in meiosis but not in mitosis. So synapses, that is pairing up of the chromosomes, homologous chromosomes, occur only during meiosis because we have their paternal and maternal chromosomes, and these chromosomes they have to move towards each other, which is called a synapsis or pairing of chromosomes. So further they have to undergo uh, crossing over with the non-sister chromatids of the non-sister chromatids of the homologous chromosomes. Well, synapsis is not a part of mitosis, but it is a part of meiosis. Therefore, moving on to the next question, question number 173. Which of the following statements is not correct? So, wrong statement we need to fetch out. The first one, pollen germination and pollen tube growth are regulated by chemical components of pollen interacting with those of the pistil. So we need to fetch out not correct one. So this is absolutely correct. The pollen germination and the pollen tube growth are regulated by chemical components of pollen interacting with those of the pistil. So the upper part of the pistil is called as the stigma. So the pollen tube has to germinate, it has to penetrate the stigma tissues and enter into the ovary. Yes, this is absolutely right. But still looking into the second statement, pollen grains of many species can germinate on the stigma of a flower, but only one pollen tube of the same species grows into the style. So this is a wrong statement or an incorrect statement. So pollen grains of many species can germinate on the stigma of a flower is absolutely wrong. The compatibility between the pollen grain and the stigma should always match. So and this match can happen only when both of them belong to the same species. So the compatibility is more with the when both the pollen grains and the stigma belong to the same species. Therefore, this is a wrong statement. But still, check out the third and fourth. Third state tells some reptiles have also been reported as pollinators in some plant species. Of course, snakes have been are also reported to be the pollinators, and insects that consume pollen or nectar without bringing about pollination are called pollen nectar robbers is also the absolutely right statement. Therefore, only option 2, that is second statement, is wrong. Question number 174. Specialized epidermal cells surrounding guard cells are called. So, when we look into the diagram of the guard cells, so they are the guard cells which are covered by, which are enclosed by bigger cells of irregular shape, which is called as the subsidiary cells. The subsidiary cells. We call this as the stoma, fibrils. So these are called as the subsidiary cells. So we have the subsidiary cells in one of, as the first option. So we'll go with subsidiary cells. So these are C-shaped cells which are called as the guard cells, and the specialized, these are the epidermal cells which surround the guard cells which are called as subsidiary cells. So the second is bulliform cells which are present in monocot leaves, lenticels which are formed during secondary growth in stem, and complementary cells. So this answer is option 1. So question number 175. Which of the following guards the opening of hepatopancreatic duct in the duodenum? So whether it is semilunar valve, sphincter of Oddi, ileocecal valve or pyloric sphincter. So semilunar valve is not a part of the digestive system at all. It is present in the valves which are present in the heart at the foot of the pulmonary artery and the aorta. Sphincter of Oddi, it is the one which is the right answer, which is the opening of the hepatopancreatic duct, which is brought about, which is called as the sphincter of Oddi. Ileocecal valve is the valve which is present between the ileum and the cecum. Pyloric sphincter, which is present at the uh, pyloric part of the stomach and the duodenum. Well, the right answer is, sorry, sphincter of Oddi is the right answer. Sphincter of Oddi is the right answer. Moving on to the next question. Stems modified into flat green organs performing the functions of leaves are known as scales, phylloclades, phyllodes, cladodes. So, stems modified into flat green organs. Example is the opuntia or the cactus. So, answer is phylloclades. They are called as the phylloclades. And we have the other options like the cladodes which are present in asparagus. The scale-like leaves we can tell which are present in asparagus, and phyllodes or nothing but the tumors. The non-cancerous benign tumors are also called as phyllodes, and cladodes I have already told. Yeah, moving on to the next question. The primitive prokaryotes responsible for the production of biogas from the dung of ruminant animals is methanogens. So methanogens are the one which are used in the production of biogas. They are present in the ruminant of the digestive tract of the cattle and buffaloes. So eubacteria are also primitive prokaryotes, or you can call them as the one which belongs to monarans. Thermoacidophiles are the bacteria which are found in hot springs, and halophiles which live in the regions of high salinity. Therefore, the right answer is methanogens. A river with an inflow of domestic sewage rich in organic matter may result in. Options are an increased production of fish due to biodegradable nutrients, drying of the river very soon due to algal bloom, death of fish due to lack of oxygen, increased population of aquatic food web organisms. So the right answer is death of fish due to lack of oxygen, because when a river is getting domestic sewage, so this domestic sewage consists of huge amount of organic matter, and because there is abundant supply of oxygen from the nature, there is an increase in the decomposers. There is an increase in the decomposers. So these decomposers will use all the oxygen for the conversion of the organic matter into the simplest form. Therefore, there is lack of oxygen, and therefore the fishes may die due to the absence of the dissolved oxygen. Moving on to the next question, question number 179. A cell at telophase stage is observed by a student and a plant brought from the field. He tells his teacher that this cell is not like other cells at telophase stage. There is no formation of cell plate, and thus the cell is containing more number of chromosomes as compared to other dividing cells. This would result in, the answer is polyploidy. The answer is polyploidy. So what we can see is, if there is no more of chromosomes, this during anaphase, if during anaphase, there is no segregation of the soon after the metaphase, if there is no splitting of the centromere and splitting of the centromere, and if the chromatids do not get segregate, what happens? Let us hope two of the chromosomes have segregated and they have started moving to the other pole. So there is one chromosome which has not undergone segregation. So this is, yeah, if this whole of the chromosome moves towards this side, so the daughter cells formed will be of this kind. So this will form one cell, this group will form another daughter cell. So the number of chromosomes in this cell will increase, and this, we call it as polyploidy because there is more than two sets of chromosomes. Here there is loss of chromosome, here there is more of chromosomes, which is leading to polyploidy condition. There is more of the chromosomes. Soon after, sorry, this is if the cell soon after